Hello students, you're welcome once again to genetic series. We're looking at a new module 4, and we're looking at population genetics. So what is population genetics concerned with? Population genetics is concerned with the transmission of genes in nature, and it studies the consequences of Mendelism in populations. This could be in groups of families or in individuals. What do we mean by Mendelism? In this case, we are looking at the effect of mutation, genetic drift, selection, migration, and others on the population. Population genetics is largely mathematical, but its theories have been found to be very beneficial in helping geneticists to understand evolution. So by population, we mean any sexually interbreeding or potentially interbreeding group of individuals. And the population may be small, might be finitely large, or infinite. A genetic population is characterized by certain attributes, the genes and the genotype frequencies. So we move. So, allele frequency. So allele frequency, also known as gene frequency, is defined as the relative abundance of a given allele in a population. Another way of looking at it is the frequency or proportion of a particular allele among all allele copies that are being considered. So the genetic composition of a population is described, therefore, in terms of its allele or allelic frequency. Let's consider a population with two different alleles, capital A and small a, of a gene. The allele frequencies are represented as P and Q, where P and Q are fractions. When you add it up, you should get 1, meaning P plus Q should give us 1. In practice, allele frequencies are estimated from what is known as the genotype frequencies, or from the genotype numbers. Now, for this to occur, we mean that all the genotypes must be easily distinguishable, although this is not always easy where there are cases of incomplete dominance. In the case of codominance, for example, the human ABO blood group, the alleles could be distinguished using chemical reagents that are specific to each blood group. Estimation of allele frequency. As an example, let us consider the MN blood type of 1,000 people in a population, and the data collected is given below. These are the blood groups. So in this population, the number of people with the MN blood group are 298, MN 489, and NN 213, making a total of 1,000. So the genotype frequencies are 0.298, 0.489, and 0.213. This is obtained simply by dividing the genotype number by the total number of people or individuals that are sampled. So if the frequency of the three possible genotypes MM, MN, and NN are represented by P, Q, H, and Q respectively, then the allele frequency of Those with blood is represented by P, while those with NN blood is represented by Q. Therefore, the frequency of the blood group is given by P plus half H, and that of NN is Q plus half H. Why half H? We should note that half of H is found in M and N respectively. So, therefore, if we are to go ahead and calculate the allele frequency of the blood group, it will be 0.298 plus half 0.489. And if you do that, you get 0.5425. To obtain the allele frequency of NN is 0.213 plus half 0.489. And if you do that, you get 0.4575. Or another way of obtaining the allele frequency of the NN blood group, having calculated that of the blood group, is to simply subtract the allele frequency of the group from 1. So 1 minus 0.5425 will give us 0.4575. Note that everything summed up to unity. So the question will be, what will happen if there were to be deaths or people emigrated or immigrated into this particular population? There are likely to be changes in the allele frequency of both the MN and the NN as a result of deaths, as a result of migration and immigration. So the above example you notice is when we are using the genotype frequencies. But we can also obtain it from using the genotype number using two different approaches. So the data that is coming up was collected from the population. It was found to be mating randomly and segregating 4K blood group alleles. This data was obtained from the work by eBay in 1988. So this is the genotype, the number, the genotype, and the number. So those with K1, K1 genotype were 25. Those with K3, K4 were 36. Those with K1, K3 were 40. K2, K3, 15. Those with K2, K4, 10. K2, K2, 8. Those with K4, K4 were 5, and those with K1, K2 were 20. Those with K3, K2 were 25, and K1, K4 were 31. So there are two approaches to estimating the allele frequencies in this this data. The first approach is what is given here. So you see that we have the genotype, K1, K1 ending with K1, K4, the number of individuals having this genotype. Here we want to get the number of alleles in each genotype, and so the genotypes are all represented. And here we want to know the total number of alleles from each of the genotypes. So you notice that in K1, K2, there are two K1, K2 represented. So the number of alleles are 2, and if we multiply this number by these 2, we get the total number of alleles at the K1, K1 locus. Here we have K1, K3, only one individual, but you notice that there's K1, K3, which means 40 of these people were having K1 and 40 were having K3. And that's the number of alleles, 1 for K1 and 1 for K3. And when the implication is that when you multiply this by this, you get 40 for K1, and when you multiply this by this for K3, you also get 40. So it's split. Similar thing happened here, because there's K2 and K3, we have K2, K4. same here is K4, K4. So the number of alleles are 2. 2 times 5 will give us 10. K3, K3, K3. 2 alleles times 25 give us 50. K3, K4. So we have K3, K4, 1 allele each. We multiply that by 1, we get 36, 36. The same thing is obtainable all the way here. So when you add up everything here, you get 215 as the number of individuals that are sampled, and add up all the K1s, the K2, the K3, and K4, you have 141, 61, and so on. And when you add up everything, you get 230. You notice that this 230 is twice this value. So, like I said before, there are 250 individuals counted. Therefore, there are 430 possible alleles. And therefore, we can now estimate or calculate the frequencies of each of the group. So for K1, the total was 141, and we divide this by the total number of alleles, we give us 0.32791. We'll do it for K2, we'll get 0.14186. For K3, 0.32791, and for K4, 0.20232. And when you add up everything, again, it sums up to 1. So the second approach, we're going to use the same data like one we use in the first approach. Now, if the number of all the genotypes are presented as K1, K2, K3, and K4 in that order, it means then that the frequency of K1 is given as below. So we have 2K1 plus K2 plus K3 plus K4, all divided by 2K1 plus K2 plus K3 plus K4. So if we substitute the values of K1, K2, K3, K4 into the equation, we have 2 times 25, which was the number of individuals with K1, plus the number of individuals with K2, plus the number of individuals with K3, plus K4. And then when we sum up everything, remember that the total number of alleles in the population is 430. So if you do 2 multiplied by whatever, all these things, you get 2 multiplied by 25. Note also that there's 1 K1 in K1, K2, which is equals to 20. That's where we got our 20 from. 1 K1 in K1, K3, that's where we got our 40. And 1 K1 in K4, K1, K4, which is where we got our 31. So if you do the calculation, you end up getting 0.32791. You also notice that this is the same value we obtained when we used the first methodology or approach. So for K2 is to substitute K2 into the equation. This will now be K2, and then K1 will go. So when you do the substitution and put in the values where they're supposed to be, you get 0.14186, which is the same as the first approach. For K3, 0.32791, and for K4, 0.20321. Again, when you add up, everything sums up as 1, which is the same as in the first method. So one of the things that came up as a result of population genetics is the Hardy-Weinberg's law. This law was collectively put together by 2 scientists who, funnily, were working independently of each other. There is a British mathematician and a German physicist. So the law states that the gene, and hence the gene frequencies in a random mating population that is not subjected to selection, migration, and mutation, will remain constant in every generation. So the key thing is that there is no selection, no migration, no mutation is allowed. Such a population, you expect that the gene and the gene frequencies should remain constant in as much as the population is mutating randomly. So let us consider a single gene locus with two alleles, capital A and small a, whose frequencies are P and Q. They are the Weinberg equilibrium frequency for the three possible genotypes. Possible genotypes are AA, homozygous dominant, A small a, heterozygote dominant, and small a homozygous recessive. Is obtained by expanding the binomial P plus Q squared, and that will give us P squared plus 2PQ plus Q squared, all summing up to 1. This principle could be applied to as many gene loci with many alleles as possible. So there are certain limitations to Hardy-Weinberg's law, because for this law to hold true, there are some assumptions or conditions that must be met. So these are that reproduction is only by sexual means, but we know that reproduction is not only by sexual means. Organisms are diploid, the population is very large, that all mating is at random, meaning that every member of the population must have an equal chance of breeding successfully, that there's no natural or artificial selection taking place in this population, that there's no migration or immigration into or out of the population. But should there be migration or immigration, the rate of migration must equal the rate of immigration. That mutation is not occurring, that all animals produce the same number of offspring, that the allele frequencies are equal in both sexes, and that generations are not overlapping. So any population that meets all these assumptions or conditions are said to be in Hardy-Weinberg's equilibrium. So we want to test Hardy-Weinberg's law, just like we tested in module 1, I think, module 2, where we tested Mendel's laws to ascertain if the Mendelian ratio of 3:1 is true, or the 9:3:3:1 is also true in certain situations. So we want to test if this law also holds true. So it can be tested just as Mendel's laws. So we'll use the data below of the population segregating at two gene locus, B, capital B, small b, and at the B blood group locus. Again, data was obtained from a work by EB, 1998. So there we have the genotypes. These are the genotypes. And we have the numbers coming up. These are the observed number of individuals with BB 104, capital B small b 356, small b 210, giving us a total of 370 individuals. So what is expected of us to test this law is to first calculate the Hardy-Weinberg equilibrium frequency. We also to estimate the expected number of individuals per allele, and then we can now ask the question as to whether equilibrium was attained after one generation of mating. So the solution, the first thing is to calculate Hardy-Weinberg's equilibrium frequencies. Let us recall that the frequency of capital B is given by P plus half H, and that was small b will be Q plus half H. So the gene frequencies, when we do the calculation for BB will be 0.155, that of capital B small b 0.531, and that of small b is 0.3. And how do we obtain it? By the number of individuals divided by the total number of people that were sampled. So therefore, the allele frequency for capital B will be 0.155 plus half 0.532. If you do that, you get 0.4205. And for small b, 0.313 plus half 0.531 will give us 0.5785. and if we should add all this, it will sum up to 1. So we have calculated the Hadwinder equilibrium frequencies. So now we want to estimate the actual number of the expected number of individuals from each genotype, not what we counted, but what should we expect. And this can be obtained using the formula that the gene frequencies square of the gene frequency times the total number of individuals. So square of this times total number of individuals. So for BB, we square the gene frequency times 670 will give us 118.47. For small b, we square this times the total number of individuals will give us 224.224. And for the heterozygote, we use this formula, which is 2 times this times this times this will give us 325.967. So these are the expected number of individuals that should have been in that population and not what was actually counted. So we now want to see whether equilibrium was attained in the population. So recall that this is the genotype and this is the observed number of people. So these are also the expected number of people from our calculation. The difference is 0 or observed minus expected, which is this minus this, will give us minus this. This will give us this, and this should normally sum up to 0. This is an indication that what we are doing so far is correct. If this value is not 0, then we'll have to go back and check if something is wrong somewhere. So the next is for us to calculate the chi-square for this using these values, and the formula is sum of the observed minus expected squared divided by the expected, which we have abbreviated to this. Sum of observed represented by O, expected represented by E squared divided by that. So when we take that into the equation, the formula rather, we'll have sum of this for BB, which is this, because this is exactly this minus the square. So bring it here, square it, divide by the expected. We bring this, we square divided by the expected, and we take this and we square it, divide by the expected. There's something wrong here. This is supposed to be minus 14.695. If you do that, the answer you get is 5.43. So the next thing is for us to actually go to the chi-square statistics, the chi-square table, and check at 2 minus 1 degree of freedom. And there the answer is 3.841. So we want to draw a conclusion from this work whether what we observe is in Hardy-Weinberg's equilibrium. Actually, two hypotheses were supposed to have been tested, or we are testing two hypotheses, the null and the alternate hypothesis. And the null hypothesis is of the view that the number we observe is actually what is expected, while the alternate is of the view that the number we observe is not what is expected. So the difference between the observed and expected number of individuals in this case is significant. And why is that? Because what we calculated, 5.43, is actually greater than the 3.842 that is on the tabulated table. Therefore, we are going to reject the null hypothesis, which is of the opinion that equilibrium was obtained in this population, and we will accept the alternate hypothesis, which is of the view that equilibrium wasn't obtained, Hadwinder equilibrium wasn't obtained in this population. So that's our conclusion. The equilibrium was not attained in this population. So this way we'll be able to test the Hadwinder law for this population. It has shown us that this population is not in Hadwinder equilibrium. So there are factors that can lead to gene frequency changes in population, and that is our next focus. So for Hadwiembeck law to be true or acceptable, this is where it's only acceptable where you don't expect much changes in the population. There's no migration, there's no mutation, there's no selection, and there's no genetic drift. But in nature, changes do however occur. People do migrate, people immigrate, people emigrate, people select, and mutation do occur naturally or artificially, and so changes occur. And when these changes occur, they definitely lead to gene frequency changes. And these changes are usually brought about by factors or forces that are external to the mating population, not necessarily within the control of the animals. And so these are classified into two. We have the systemic, things like migration, mutation, and selection. And then we have the non-systemic, like random drift or chance. So the systemic forces, migration, mutation, selection, occur. In fact, they operate naturally, although they can also be enforced, but they do operate naturally. And so animals and plant breeders use this to bring about changes in the phenotypic value of individuals. Changes in the phenotypic values are primarily due to changes in the frequency of the genes governing or controlling such characters of interest. So let's take a look at systemic forces. Migration. Migration, as we know, is a movement of individuals or populations from one place to another. Seasonally, if we are very observant, we see that birds that we see in certain seasons are not seen in certain seasons. Fish, butterflies, and other animals seasonally move from one location to another. Locally here in Nigeria, one of the easily noticeable migration is a yearly migration of Fulani pastoralists with their cattle from the southern part to the northern part during the dry season, during the wet season, and from the northern part to the western part during the rainy season. Yes. Now, such movements usually we introduce different genes into the new mixed, what we call the mixed population, and because of such movements and introduction of new genes, there will be gene frequency changes in the new population when compared to the old population. So there are many reasons for why migration occur among animals, particularly things like deforestation or afforestation, flooding, rise and fall in ocean tides, changes in weather condition, changes in ocean current, food scarcity, availability, warfare, particularly in case of human migration, and any reason that can make an animal to move. Some migrate to go to particular parts of the world for mating and nurturing of their young ones before going back to their natural habitat. So, because of this migration, we increase genetic variability of a population as a result of introduction of new genes into the old population. So when plants and animal breeders purposely introduce new genes into a population through outbreeding, we also refer to this as migration. So we want to obtain an expression for calculating the expected change in gene frequency as a result of migration. So let's look at population the size, the frequency of the gene in that population. So what we have here in the native or original population, the size represented by NO, O meaning original or native. So the frequency is QO. The migrant represents small m, NM, the frequency is QM. When mixed together, we have NO plus NM, which is very by simplicity. So for us to get a proportion of immigrants or those who migrated into the mixed population, this is the same thing as the migration rate, and it's given by the expression that M is equals to NM divided by NO plus NM. So the population of the natives in the mixed population is N1 minus M, which is the same thing also as NO divided by NO plus N. The frequency of the gene in the mixed population is estimated as a weighted average for the frequencies in the native and the migrant population before integration. So thus the Q, which is the genes frequency of gene in the mixed population, is given by 1 minus N, which is the bracket Q, which is the migration and the proportion of the non-migrants plus MQM. So the expected change in G frequency due to immigration is also given by Q is equals to Q minus QO. And remember that Q is 1 minus MQO plus, if we bring this in now, this will be this minus this. So the change in gene frequency is M. When you cancel out what is not supposed to be there, you get M bracket QM minus QO. So you can use these equations, these formulas to do this assignment. So the assignment says that 10 land rights pigs were introduced into a population of 50 indigenous pigs for the purpose of upgrading them. So this, the initial number, population, these are the migrants. So if the frequency of the stress gene presented by N in the land race and the indigenous pig were 0.75 and 0.2 respectively, we are expected to calculate the expected change in gene frequency or in frequency of the gene in the local population as a result of the introduction. So if you go through the processes from the first formula to this one, you'll be able to solve this assignment. Mutation is another systemic factor that can lead to gene frequency. And mutation is any heritable change in genetic material. So being heritable means that whatever mutation takes place, it is passed on to the next generation. Mutation can be brought about by processes such as deletion, addition, inversion, duplication, or substitution of genetic bases. It may also result from structural changes in the chromosome. Mutation can be naturally induced or artificially induced, and it happens spontaneously. So when it is induced, we do it using chemicals such as nitrous oxide, amine acid, muflagine, or radioactive exposure. Mutation is a very slow process of the order of 10 raised to minus 1, 10 raised to minus 6 mutation per generation, and it makes it quite slow process. And where it happens most is usually referred to as the hotspot. That is where it happens most frequently, and it is a very important evolutionary tool. It provides raw materials for natural selection. Mutation can be used to create new genetic variation, and genetic variation are the basis for selection. So we want to have a look at some chromosomal aberrations that will lead to mutations. So chromosomal aberrations, while not changing the chromosome number, it does alter the genetic makeup of the individual. And mostly it comes from breakages in the structure of the chromosome. So these are the common types of chromosome aberration. We have inversion. And what happens in inversion is that when a chromosome breaks off, this chromosome is breaking off at this point, and it rotates 180 degrees, and then it rearranges itself within the same chromosome. So you can see the chromosome is breaking up at this point and also at this point. So when there's a 180 degrees rotation, complete rotation, this joins here and this comes here. So the new chromosome is looking at ABE and AB and CDFGH, which is what we have here. So another chromosomal aberration is duplication, and duplication is when a rearranged chromosome contains extra copy or copies of a given segment. That is, in this case, there's a repetition of the chromosomal material. So this is an example of a duplication. So we have this original chromosome, homologous chromosome A, B, C, D, E, F, G, A, B, C, D, E, F, G. So because of a duplication of a C, what we are now having is A, B, the C is appearing twice, D, E, F, G. Deletion is another chromosomal aberration, and this is when a segment of a chromosome is lost due to breakage of or union of the chromosome. And when there's deletion, there's usually a loss of chromosomal material, and that has great implications on the individual. So in this example here, we have a chromosome A, B, C, D, E, F, and here the E has been deleted, and what we get is A, B, C, D, F. The E is no longer there. So there's also translocation, and this basically involves interchange of chromosomal parts between non-homologs. That is, the chromosomes involved here are not homologous. There's transfer, therefore, of genetic material of one of the chromosomes to the non-homologous chromosome, and this is exemplified here. These are non-homologs. You can see they have different letters. So when there is that happening, we end up having A, B, C, D, E, Z. Okay, and then A, B, C, you see D, E, Z, and here you have U, E, U, W. Now have your X, Y, and F, G comes here. So this moves up, and this 2 takes its position. So if there are n number of A genes in a population and only small n of it changes to small a in one generation, then we can also obtain the mutation rate, which is given by F, which is n, that is the change from the original, the total number of capital N. And if the initial gene frequency of A is given as PO, then the gene frequency due to the mutation of A is given as FPO, frequency of PO. And to obtain this is the mutation rate multiplied by the initial gene frequency. Okay? Now, therefore, we can also obtain the new gene frequency of A, which is PO minus FPO. That is the initial gene frequency minus the gene frequency due to mutation, which is this. Then we can also obtain a change in gene frequency using this formula, which will give us this at the end of the day. So let's look at selection. Selection is natural and artificial. Um, Selection happens even when we want to get married, we do selection. That is artificial selection. So selection is a keyword when we talk about the concept of survival of the fittest. This concept arose as a result of selection, nature's ability to select those that will survive to reproductive age, and that is naturally, or when artificially we select animals or plants that we feel are fit enough to survive to reproductive age. So normally the average phenotypes, the heterozygotes, are fitter and they are more likely to survive than the homozygotes. And the reason for this is because of the effect of hybrid vigor, or what is known as heterosis. So it means that natural selection is directed against extreme phenotypes, the homozygote dominant and the homozygote recessive. So the heterozygotes are more likely to survive in any population or in any extreme situation than the homozygotes. Now, artificial selection, just like mutation and uh, migration, could be used by plant and animal breeders to handpick individuals that will be used as parents to produce the next generation of animals and plants due to their fitness. And selection is based on measurable traits. Um, However, it may not be that easy selecting animals directly based on the genes which they govern or control, or the economic traits that they control, because such traits are usually controlled by many genes. But there are exceptions, and that is when quantitative traits are being controlled by few genes. Okay, so we'll be looking at two important terms as far as selection is concerned, and these are fitness, represented by the letter W. So what is fitness? Fitness is concerned with the contribution of an offspring to the next generation. So it is a function of the offspring's ability to survive to reproductive age and also the number of offspring that it can produce. So individuals that survive to reproductive age are said to be fit, and the reverse is also true for those that do not survive to reproductive age. They are not fit. So another concept is selection coefficient, represented by capital S. And this measures the strength of selection. It's defined as a proportionate reduction in the contribution of a particular genotype relative to the most favored genotype. Thus, if the most favored genotype has a fitness 1, then the relative contribution of a genotype that is selected against is 1 minus S. For a letter allele, the fitness is 0. And why is that? Because they have very slim chances of survival. These kind of individuals will not survive to reproductive age. So fitness is 0. So the selection coefficient then becomes 1. Therefore, its selection coefficient is 1. That is 1 minus 0. Get 0. So we want to show the relationship between fitness selection coefficient using this example. So we have a genotype. Okay, these are the offspring produced by the genotype. Uh, these are the offspring having the different genotypes. So remember the relative fatness W, we say it is the number divided by the most fit. So this divided by this gives us 1, this divided by this gives us 1, this divided by 100 gives us 0.3. and the selection coefficient is 1 minus relative fitness. So 1 minus 1 is 0, 1 minus 1 is 0, 1 minus 0.3 is 0.7. So this is how we can go about calculating relative fitness, number of progeny produced divided by the number of progeny produced by the most favored. We had said that the heterozygotes are usually the most favored. They're able to survive extreme conditions, and selection coefficient is 1 minus this. So the most favored genotype is AA. Finally, we take a look at chance. Chance, like the other three we have looked at, also leads to gene frequency changes. The only thing here is that while the gene frequency changes can be predicted in their amount, but the direction in which the changes may occur is very difficult to predict. So because of this, we say it is dispersive in nature, but good enough it occurs mostly in small populations. And in such small populations where gene frequencies can fluctuate randomly from generation to generation, it may lead to fixation of a gene or a total loss of such a gene in that population. So when this happens, useful genes, when present at very low frequencies, may become lost totally from the population. Meanwhile, deleterious recessive genes, however, may become fixed in that population, leading to reduced viability. So the amount of gene frequency changes due to chance is also given by this formula, where Q is the initial gene frequency, okay, and N is the total number of individuals. So chance will be greater with smaller sample size or population. So in closing, give you this assignment. So given that the initial gene frequency of a population for a trait is 0.6, what is the variance due to chance when the number of individuals in the population is A, 10, B, 5, and 3, and C, 100? So go ahead and do this, and you'll be able to obtain your correct answers. So thank you for listening, and what next? So keep your eyes focused for the next video on this series, the genetics series. And if it has been beneficial to you, go ahead and click on the subscribe button and subscribe so that you get the next video in the series. Share with a friend and drop a comment. See you on the other side.